People like a minute. Started. It's my real pleasure to introduce Nausicaa Arnault. She is an assistant professor at the Molecular Cellular Developmental Biology Department in Boulder at our sister university, University of Colorado Boulder. She got her PhD at the Institut Curie in Paris. And if you have not been to that institute, I highly recommend it. It is the most fascinating museum around. It's like 2 rooms, it's teeny, but you will learn so much about Marie Curie and her daughter and the amazing women that they were. Oh, and I should get back to Nausicaa. And there at the Institut Curie, she studied telomere biology and replication. And then she did a 2-year postdoc in Belgium, and she again worked on telomere chromatin before she joined the Salk Institute as another postdoc. She did use telomeres as a tool to study DNA double-stranded break repair. She joined the University of Colorado Boulder in 2018, and she's been doing a lot of understanding of DNA repair, still with the focus on telomeres. But what she'll be talking about today is using CRISPR-Cas9 screens to study DNA repair and the function of Ape2 and alternative end joining. So without further ado, Nausicaa, take it away.
Thank you, James. Thank you for the nice introduction, and thank you for being here today. I'm going to show.
And while you're doing that, Nausicaa, let me mention one thing. It's really nice if everyone asks their own questions. So probably the best way to do that is just put your in the chat. Just put your name. In other words, almost get in line for a question. And at the end, I'll call on people in order, and you can ask your own question and just turn on your screen so it's a little more real, so people can see you, and then ask your question. That way, you can get it back and forth. And if I'm sure Nausicaa would entertain questions during her talk as well. And in that case, I think you should just break in, because I won't be checking the box during the talk. Anyway.
Yeah, as I said, I use Keynote, so as soon as I switch to full screen, I can't see you, I can't see the chat. So if you want to interrupt me, just talk. So as James said, I started my lab only 2 years ago, so it's not going to be a 50-minute seminar, so we'll have plenty of time for questions. But I'm very happy to talk about this story because we're very excited about it. So my lab studies both telomeres and really just the telomere biology and double-strand break. So we also use telomeres as a tool to study double-strand break, and I'm going to explain that in my talk. So first, why study DNA repair in the relevance for cancer? So as you all know, genomic instability is a hallmark of cancer cells. And if you have mutations in DNA repair genes, this is really the best, the easiest way to have genomic instability and to accumulate more mutation faster. And so this is not surprising that there's a lot of DNA repair genes that are mutated in cancer, and that mutations in DNA repair genes, individuals who have mutated DNA repair pathway have a high prevalence in cancer. So then studying and understanding DNA repair is first necessary to understand how tumorigenesis process happen, because of course the failure to do proper DNA repair participates in the early stage of tumorigenesis. It's also important in terms of diagnosis because, as I said, DNA repair mutations lead to cancer predisposition, and so the more we identify new genes or new gene functions, the better we can identify this predisposition. And one of the best examples is homologous recombination. So you all know about BRCA1, BRCA2 cancer that lead to mutations that lead to a high prevalence in cancers. BRCA1, BRCA2 are part of homologous recombinations, and the more genes that we know in this pathway, the more we're able to diagnose the fact that people will have high cancer prevalence. And finally, understanding DNA repair is important to treat cancer because a lot of cancer therapies are either based on inducing DNA damage, either by drugs or by radiation, but also some inhibit directly DNA repair pathways. So with that, I'm going to move to double-strand breaks, which is one type of DNA lesion. It's rather rare, but it's considered the most toxic type of lesion. And this is the type of lesion that you create when you do radiation therapy. And double-strand breaks, as we usually represent them like that, can be repaired by two main pathways: non-homologous enjoining, that just brings the two ends and ligate them together, and that is considered rather toxic, or homologous recombination that occurs only during S and G2, and that uses the sister chromatid to serve as a template for repair. However, there's a third pathway called alternative enjoining, and alternative enjoining is going to use microhomologies around the break site to repair DNA. It's not very clear what is how important alternative enjoining is in regular cells. We know that it's a backup pathway. If you don't have either NHEJ or HR, you're going to activate alternative enjoining. But some studies actually suggest that it is occurring even in cells that do have a proficient NHEG and HR. Okay, so these are double-strand breaks as we often represent them. However, these double-strand breaks very rarely occur in cells unless cells have irradiations, or you have cells that induce them physiologically, either because it's during meiosis or VDJ recombination. So the most frequent, the overwhelmingly frequent DNA double-strand break that occurs in cells that are not challenged are what we call replication-induced double-strand breaks. And that's when you have a single-strand break that during replication, at the replication fork, gets converted to a double-strand break. And this is very different because it should not be repaired by an HEG, because you can imagine that if you just fuse the ends like that, that leads to a very toxic structure. So this is why NHEG is actively inhibited at this type of double-strand breaks, and HR is the main pathway. Now, as I said before, HR is frequently mutated in cancer. All the BRCA1, BRCA2 cancer, they cannot do HR. And in this case, they actually completely rely on alternative enjoining to repair all the double-strand breaks that occur during replication. This means two things. First, this explains why there's such a prevalence of cancer in HR mutated individuals, because alternative enjoining is a toxic repair. The other thing it means is that we have a way to treat these cancers, because if you now target and inhibit alternative enjoining, the cell is no longer able to repair this double-strand break that occurs during replication, and they die. And so this is what happens when we treat HR deficient cancer with PARP inhibitors. So PARP plays a role in many different DNA repair pathways, but it is involved in alternative enjoining. And so if you suppress PARP, you're going to suppress alternative enjoining, and this is lethal. The problem with PARP inhibitors is that there's a lot of resistances to PARP inhibitors. So currently there's a really large effort to try to find novel targets. A lot of companies are trying to develop inhibitors against Pol theta, which is a protein coded by POLQ gene that is part of the alternative enjoining. However, the more we know about the alternative enjoining pathway, the more targets we could get. potential targets. And so that's why in my lab, we decided to try to identify novel genes in the alternative and joining pathway, because this is still a very poorly defined pathway. And for that, we wanted to use the synthetic lethality between HR and alternative and joining. So the idea is simple. If you take either HR proficient or HR deficient cells and infect these cells with a genome-wide knockout library, any guide RNA that is going to target an alternative and joining gene is going to be lethal in the HR deficient cells, but not have any effect in the HR proficient cells. So this is what we did. There's a lot of labs who actually looked at what is synthetically lethal with loss of BRCA1 or loss of BRCA2. What we really wanted to do was to identify what is lethal with the HR pathway in general. And so for that, we needed isogenic cells with different mutation in the HR pathway. So we decided to look at two different genes, BRCA1 and PALB2, because they are rather, they play roles in different steps in the HR pathway. And so we used a cancer cell line that is HR proficient. And with CRISPR-Cas9, we knocked out either BRCA1 or PALB2. So that was the first step of our study, was to generate the cell lines. So we did that, and each time we isolated a single clone because we did not want to introduce the library into, well, we wanted to introduce the library in a homogeneous population, so a clonal population. So this is the knockout clone that we selected for BRCA1. You'll see that it's the guide RNA is really early in the gene, in the protein. We have a nice knockout. Same for PALB2 here, with the knockout that is here. This is unspecific band. And then we confirmed that these clones were indeed HR deficient. And there's two main ways to see that it's HR deficient. The first way is to look whether the cells can form RAD51 foci. So at the very end, almost the very end of the HR pathway, there is invasion of the sister chromatid with RAD51 filaments. So if you have an HR compromised cell line, you should not be able to form these RAD51 filaments. So we irradiated all these cells, the normal HT1080, the BRCA1, the PALB2 knockout. These cells form gamma H2AX foci. However, they can no longer form the RAD51 foci, which indicates that indeed they are mutated, deficient for homologous recombination. Then we also control that they are sensitive to PARP inhibitors. So remember, PARP inhibitors will kill the alternative and joining pathway. And so cells that are HR deficient should die from that. So we measured that by clonogenic assays, as well as by following cell growth over time. And as you can see, when we use PARP inhibitors in HT1080, there is not a large effect unless we use very high concentration of drugs. But if we do the same in BRCA1 and PALB2 knockout, then the cells are way more sensitive. Okay, so now we had our isogenic cell lines that are either HR deficient or have two different HR knockouts. We infected all these cells with a genome-wide library and counted 10 population doublings. So these cells don't grow at the same speed, so we made sure that they really went through the same number of cellular division. And then we looked first at a comparison between the BRCA1 knockout and the HT1080. And so all the genes that you see here are synthetically lethal with BRCA1, meaning they are lost in the BRCA1 knockout, but not in the parental HT1080. And then we did the same for PALB2. And as you can notice, there are some proteins, some genes that are found in both genes, but a lot of them are actually specific. So for example, BRCA1 knockout is synthetically lethal with a lot of genes that are involved in the Fanconi anemia pathway. This is not true for PALB2. So anyway, what we were interested in was not looking at the synthetic lethality of these genes individually, but together, what is common to what is synthetically lethal with the HR pathway, which the prediction that we would find the alternative and joining gene. So we did that. We basically plotted the depletion p-values of BRCA1 versus wild type on PALB2 versus wild type. And you see that there are some way less genes that really pop out of these screens. And in blue are all the genes that are involved in alternative and joining. Except for one gene that is known to be part of alternative and joining, that is FEN1, and I'm going to show you why we don't find FEN1. This is very simple. FEN1 is the flap endonuclease. It's required for a general genomic DNA replication. And so if you suppress FEN1, this is lethal not only in BRCA1 and PALB2 knockout, but also in regular cells. So that's the guide RNAs that are found here. They are lethal in all cells, and that's why we cannot identify it in our screen. On the other hand, we have all the other alternative and joining genes, which really for us was a good sign that this was the perfect technique, a very powerful technique to identify alternative and joining genes. So beyond these already known genes, we identified 3 genes: Apex2, CYP2A, ALC1. CYP2A is a phosphatase. It's involved in cell cycle regulation and in DNA damage and DNA repair. ALC1 is a chromatin remodeler that was shown to be involved in DNA repair, and Ape2 is an abasic site endoexonuclease that is involved in base excision repair. Sorry. So, first thing was to confirm that these 3 genes were indeed synthetically lethal with BRCA1 and PALB2. So we took again our HT1080 parental cells, BRCA1 knockout, PALB2 knockout, and we introduced guide RNAs against these 3 genes and against POLQ as a positive control, because we know that POLQ is involved in alternative and joining. As you see, the depletion of these genes have very little effect on cell confluency and on colony formation in HT1080 cells, but it is lethal in BRCA1 knockout and PALB2 knockout. This is the colony formation, and that's the count of cell death by annexin 5-PI. So there's a very clear effect that is pretty much the same as what we see in POLQ. So from here that we confirm that Apex2, ALC1, and CYP2A are synthetically lethal with the HR pathway, the next question was, of course, are they involved in in alternative and joining? There's 2 ways to measure alternative and joining activity. One way is to use DNA repair reporters, which we are doing on the side, but these DNA reporters have a lot of weak points. So we went directly to the gold standard, which is using chromosome fusions, telomere fusions. So, and I'm gonna explain you how it works. So you know that telomeres are the ends of chromosomes, and one of the roles of telomeres is to solve what we call the end protection problem. What that means is that because they are the end of linear chromosome, they basically look like double-strand breaks. So they need to inhibit all the DNA damage signaling and the DNA repair pathway to avoid fusions. And they do that by recruiting a specific complex called the sheltering and forming a T-loop, so a loop in which the telomere folds and hides basically the end of the chromosome. So the different proteins in the sheltering complex inhibit different DNA damage signaling and different repair pathway, which means that by depleting specific proteins, we can reactivate specific pathways. And this is really interesting because if you look at the repair and intrachromosomal double-strand breaks, you're always going to have the different pathways that are competing. So if you see a reduction in alternative enjoining, you don't really know if it's a reduction in alternative enjoining or, for example, an increase in NHEJ. With this telomere fusion, we really know exactly what we're looking at because we can just induce one pathway, but yes, keep the other one suppressed. So how does that work? TRF2 is the inhibitor of the ATM signaling and non-homologous enjoining. So if you suppress TRF2, you reactivate ATM signaling and you reactivate NHEJ. And that leads to these spaghetti chromosomes where all the telomeres have fused together. If you now also inhibit ligase 4, for example, that is required for NHEJ, you still have this ATM signaling, but now you inhibit NHEJ and you suppress fusions. This has been used to identify NHEJ proteins in the past. But here we were interested in alternative enjoining, and alternative enjoining is inhibited by TRF2, TRF1, and Q70, Q80. So if instead of inhibiting ligase 4, you suppress Q70, Q80, then because Q70, Q80 is part of the NHEJ pathway, you still suppress the NHEJ pathway, so you don't have any NHEJ mediated fusions, but you also reactivate alternative enjoining. And then you have fusions that are mediated by alternative enjoining. There's less fusions because we still have TRF1 that inhibits a little bit alternative enjoining. If you want to have these spaghetti chromosomes, you need to inhibit everything, but it's not very easy to do this triple knockout. So we chose to just inhibit TRF2, suppress TRF2 and Q70, Q80. This is not possible to do in human cells because Q70, Q80 knockout is lethal in human cells. However, it is not lethal in mouse cells. So what we did is take MEFs that have a TRF2 fluxed allele and a CRI-ERT2, which means that upon addition of tamoxifen, upon addition of tamoxifen, we can induce TRF2 depletion. And then we use CRISPR-Cas9 to do a Q80 knockout in the cells. And again, we selected a single clone. So that's different clones. We selected clone 7 and we tested that the fusions were working. So if you don't add tamoxifen, you have no fusions. If you take Q80 wild type cells, so you just inhibit TRF, you just suppress TRF2, you have fusions, and these fusions are sensitive to NHEJ inhibitors, but not to alternative enjoining inhibitors. If now you do the same, but in cells that are Q80 knockout, you induce fusions. As I told you, you induce a little bit less fusions, but now these fusions are no longer sensitive to NHEJ inhibitors. Instead, they're sensitive to alternative enjoining inhibitors. So we really have a nice tool here to now look at our different candidates and see whether if we inhibit our candidates, are we going to reduce these alternative enjoining fusions? So that's what we did. We took these MEFs that are Q80 knockout, TRF2 flux minus, and added tamoxifen and a guide RNA against different proteins. So that's a non-targeting guide RNA. You have the same profile. Now, when we used a POLQ, 2 different guide RNAs against POLQ, which is an alternative enjoining gene, you see that very nicely we suppress all the fusions. The alternative enjoining inhibitors has no more any effect on that because the level of fusion is already really low. And we were very excited to see that when we do the same with 2 different guide RNA against Apex2, we see the same level of fusion reduction as with PolQ, which really means that Apex2 is absolutely required for alternative enjoining. And this is totally novel. Now we do the same with CYP2A and ALC1, and clearly these two have no effect on alternative enjoining. So these 2 genes are not involved in alternative enjoining. We have some hypotheses why it is synthetically lethal with HR, but now on we'll focus on Apex2. So we confirm that Apex2 is suppressing alternative enjoining. It is not suppressing the fusions in Q80 plus knockout, so the NHJ fusion. So it's really just a function in alternative enjoining, not in NHJ. So that leads me to my first conclusion, is that Ape2, so the protein encoded by Apex2, plays a critical role in alternative enjoining. And the next questions were, which domains of Ape2 are involved, and what is the mechanism of action? So Ape2 has 3 main domains: the endo, exo, and phosphodiesterase activities here, a pCNA interacting domain, and a zinc finger DNA binding domain. I told you that Ape2 is involved in base excision repair, and in base excision repair, all these domains promote the activity of the protein. They all are necessary for base excision repair. So we wanted to know if it was true as well in for alternative enjoining. So we create mutants in mouse mutants for Ape2, so that's the wild type, 2 different mutations that lead to catalytic death, so it suppresses both the endo, the exo, and the phosphodiesterase activities of Ape2, a mutant that does not have the PIP domain, and a mutant that prevents the DNA binding of the zinc finger. And we express that in our MEFs that have the Q80 knockout, the TRF2 inducible knockout, so we can do telomere fusions. And they also have, a guide RNA against Ape2. So they are Ape2 knockout. And what we're looking at is a rescue of the alternative enjoining activity. So here with an empty vector, there's no rescue, there's almost no alternative enjoining fusions. We express a wild type construct, we rescue the fusions. With the PIP and the zinc finger mutants, we still rescue. So these 2 are not required for alternative enjoining. And when we use a catalytic death mutant, they do not rescue anymore. So really in alternative enjoining, it seems that only the catalytic domains are required, not the PIP domain, not the zinc finger. Then that leads me to my next question, which is what is the mechanism of action? And well, we don't have the answer. Unfortunately, we're still doing the experiments, but I'm gonna explain you what is our hypothesis and what is our strategy. So here I've depicted the alternative enjoining pathway. So if you have microhomologies here, the first step is a resection that leaves some 3' overhangs here. This resection is done by CTIP and mRNA. And then Pol theta, the gene PolQ, is going to remove RPA that binds here to enable the annealing of the microhomologies, which leads to 3' flaps. These 3' flaps likely need to be removed before Pol theta can polymerize here and fill in the single strand DNA before XRCC1 ligase 3 ligate the repair, the break. We do not know what is doing the 3' flap trimming. In yeast, it's known that it's ERCC1 XPF. So it was proposed that was the ERCC1 XPF also in mammals. However, when it was tested, it doesn't seem in mammals to play, in higher eukaryote, to play a role in alternative unjoining. So there's still a big question on what removed these flaps. And because these are 3' flaps and Ape2 with 3', 5' exonuclease, we think that this is what Ape2 is doing, is removing these flaps. Now, how to test that? One way to test that is that if Ape2 is required to remove the flaps, then if you don't have the flaps, you should not need Ape2. And you don't have the flaps when the microhomologies are directly at the end, just flanking the double-strand break. In that case, you have resection, annealing of the microhomologies, and no flaps. And so in that case, you should be able to continue with alternative unjoining even though you don't have Ape2, if Ape2 is indeed required to remove these flaps. So how to test that? Well, we basically need to test the activity of Ape2 at that type of double-strand break versus that type of double-strand breaks. So what we did is to create a specific reporter for that. So in that case, we expect that if Ape2 is required to remove the flaps, well, if we do have flaps, we won't be able to have alternative unjoining in this setting if we don't have Ape2, but we should still be able to have alternative unjoining without Ape2 at this type of break. Okay, so here's the system that we set up. We made an M-cherry cassette that is interrupted by a sequence that has a stop codon and microhomologies. So these microhomologies belong to the M-cherry. So we express a guide RNA, and when we express Cas9, that is going to lead to a double-strand break here. If there is repair by alternative unjoining, there's going to be annealing of these microhomologies, trimming of the flaps, and polymerization and ligation. And that leads to reconstitution of the M-cherry gene. So in that case, the cells become M-cherry positive. So by measuring by flow cytometry how many M-cherry positive cells we have, we know how many cells did alternative unjoining here. And then we have a second reporter that is basically the same, except in this case, the guide RNA will lead to a cut that leaves the microhomology directly around the break. And so when these microhomologies are annealing, there's no 3' flaps. And so in that case, we expect that without Ape2, we should still be able to see M-cherry positive cells here, but not here without Ape2. So in conclusion and in summary, we've used CRISPR-Cas9 and a dual screen, so a comparison of 2 different screens to identify genes that are synthetically lethal with the whole HR pathway. And that led us to identify Ape2 as an alternative unjoining factor. And we're still deciphering the exact mechanism. So why is that important? Well, first, in terms of knowledge, we still need to know more about alternative unjoining. It's still poorly characterized, so that's important. More than that, because we're looking for targets to kill cancer cells that are HR deficient, we think that Ape2 could be used as a target and could be inhibited in HR deficient cancer cells. So now we're going to look at whether inhibition of Ape2 can be used in PARP inhibitor resistant cells. And finally, this shows how when you look at synthetic lethality between genes, you can identify new functions of known genes, because Ape2 was already known to be a DNA repair protein, but when you do this screen, you clearly see that it is clustering with alternative and joining. So it shows a new function. And so what we're doing now is doing the same for all DNA repair genes. So we're basically crossing all DNA repair versus all DNA repair knockouts to be able to map all the genetic interactions between all these factors and see what clusters with what. And we think that with that, we're going to identify new functions of already known genes. With that, I would like to thank my lab. So this is mostly the work of Hubert Fleury, very talented postdoc in my lab, although a lot of people have contributed, including the undergrads who are counting chromosome fusions at home. And I will be very happy to take questions. And I can see you now.
Great. Thanks so much, Nasser. That was fantastic. It's a really interesting project. I know I saw Ben Bitler had a question.
Yes. Hi, Nassika. Thank you
Hi.
for that wonderful presentation. I have two questions, comments. My first comment is, my lab is very interested in PARP inhibitor resistance, and we have a panel of 8 matched sensitive and resistant PARP inhibitor resistant cell lines. So I'd love to talk with you offline about that. My question was related to your initial knockout of PALP2 and BRCA1. You saw differential responses in olaparib to PALP2 knockout versus BRCA1 knockout, whereas BRCA1 was more detrimental. Can you comment on why you think that might be?
On the PARP inhibitors?
Yeah, and your dose response.
Yeah. So the thing is, BRCA1 is going to do different things in the HR pathways. So there's one part where it helps PALB2 loading RAD51, and there's one part where it actually controls the end resection. So I think this is one possibility, is the fact that because if you don't have BRCA1, you don't have the control over the end resection, then you're even more sensitive to PARP inhibitors. Now, that being said, it could also be due to the fact that BRCA1 is likely to play a role in other pathways, and PARP is playing a role in different pathways. So that could explain why. We don't see always the same exact response. So for example, POLQ knockout has a stronger effect on the PALB2 versus the BRCA1. So I think it all comes to the fact that all these proteins are very redundant and play roles in different pathways, and there's never just one reason why it's synthetically lethal. I don't know if I answered.
Nasser, before I jump to others, I have a follow-up to that. So I know, at least for breast cancers, and Ben may be able to comment on ovarian cancers as well, that there are cancers that don't show known mutations in the HRR pathway, and yet show, I forget whether it's HRR deficiency functional assays or just show PARP inhibitor sensitivity. But either way, would it be worthwhile to kind of go through a panel of these cancer cells and see if your pattern with the the Ape2 sort of dependency is similar?
Yeah, that would be really interesting, because that would tell us whether these well, that would tell us different things. But there's two possibilities when you say these don't have any mutation in HR. Well, they don't have any known mutation.
Yeah,
but
exactly.
still possible that there are some HR genes that have not been identified. And that's why we've also done a PARP sensitivity screen to try to identify new HR proteins. And I forgot what I was going to say next. Yeah, no, the other possibility, of course, it's that it's involved in another pathway. One of the questions we're asking, for example, for Apex, because Apex is also involved in base excision repair, like PARP, that is involved also in base excision repair, there's really a possibility that the synthetic lethality is a mixed thing. You don't have BER, so you create more replication-induced double-strand break, and then you can't repair them. So we're going to try to see exactly why is this synthetic lethality? Is it really just due to the function in alternative enjoining, or is it both? One element of response of that is if you look at the domains. So I've told you that the PIP domain and the zinc finger domain are involved in BER, but not in alternative enjoining. If you do the synthetic lethality with PARP, if you don't have the PIP and the BER domain, the cells are not sensitive to PARP inhibitors. So it does seem like the function in the alternative enjoining is more important, but it could still participate.
Yeah, okay. And it looks like, Anthony, I don't think you actually had a question there, right? Jump in if you did before I move to 1010.
Not really.
Oh, okay, so go to 1010.
Hi, that was great. I've heard bits of it, but it's good to hear the whole story all at once. So the question I have is, you can test the role for FEN1, which is a flat nuclease, because it was already lethal regardless, right? So do you think that FEN1 might have a role still, because it's a 5' 3' nuclease, whereas you're thinking that Ape2 is a 3', 5' nuclease?
So,
or do you think only Ape2 is needed?
No, I do think, so interestingly, FEN1 was found synthetically lethal in other screens. So apparently, I guess it really depends what is your time frame of your screen. So some people manage to get it not killing wild type cells, but killing HR negative cells. Where is my pathway? Yeah, I didn't put it here. What I think is that because it's really a 5' exonuclease, when you have POLQ filling the gap here, it might goes a little bit beyond just the single strand DNA and create a flap here that then FEN1 would be required to remove. So that is why really I think FEN1 plays a role here rather than there.
Got it. Thank you. And may I ask a quick follow-up? Is that if you think that Ape2 DNA binding domain is not required, how is it binding DNA?
That's a question in the R1 that you're going to review. So we're going to look at how it's recruited. So first, there's the possibility that there's a compensation between pCNA recruitment and the zinc finger. So we're going to mutate both. Maybe when we mutate both, we're going to lose the rescue. So that's one possibility. A lot of proteins are recruited by parylation. Mm-hmm. However, I did not find in Ape2 any domain that is known to bind to parylation. So I don't think it doesn't look like it's going to be parylation. It could be something else. We're going to see whether Ape2 can bind directly to Pol theta. Mm-hmm. And we're going to basically do mass specs in cells that are HR positive, HR negative, and with regular Ape2 bind to a turbo ID, or Ape2 without the zinc finger and the pCNA binding domain, to see if there's something obvious that comes up that would recruit Ape2 to the site. But right now we don't know.
Okay, thank you.
Now, Sika, what is parylation?
Oh, it's what the PARP is doing. Adding the
okay. I'm sorry. I was hearing in my head PER. Now
no, sorry.
Yeah. I just pronounced it a little different. That's fine. Okay, that makes sense. And I think Jay is probably up next.
Hey, Nausicaa, great talk. I had a couple questions thinking more about Apex2 from the AP endonuclease side. So is there biochemical evidence that Apex2 actually acts on A basic sites? At least I have a couple of related things that you could just think about. So that's my main question. But then, from what I know, at least from yeast, I mean, Ape1 is it's basically acts on an A basic site, and it's technically an endonuclease, but it's kind of a crummy one. It basically cuts off one and then it's done. So it's more of like a distributive endonuclease, if that's what you want to think about it. And this all leads to my, the idea is, do you know, or do you think there might be an A basic site in this pathway somewhere? I can imagine like cytosine deamination, and then uracil gets popped off, and then you've got a way for Apex2 to get in.
So the answer to your last question is no, I don't think. And the reason for that is that although Ape2 is still classified as AP endoexonuclease, if you look at the site that binds and the catalytic activity, and you compare, basically it's really conserved between AP1, AP2, and the bacterial exo3 exonuclease. But if you look at the catalytic site, AP2 is different from AP1 in what really gives AP1 this abasic site activity. So AP2 has actually a very weak abasic site endonuclease. It is not clear whether it keeps a strong endonuclease at things that are not abasic site, but it has a much stronger exonuclease than AP1. So I would bet more on the exonuclease activity of AP2, but that is definitely something that we need to test with biochemistry.
Thanks.
Great. And let me see. Ben, you had a comment there that may be good for everyone to hear about the depth map.
Yeah, you can have BRCA1 methylation without mutation in the pathway. So that's answers maybe one of your questions, James, but one of your questions about looking at other repair components and synthetic lethalities, depth maps data can potentially answer that. Olaparib was included, so some of that PARP inhibitor stuff is there. My question was related to the telomeric fusion. Part of PARP inhibitor response is now starting to be attributed to activation of the CRISPR-Cas9 pathway. Do you know, with respect to AP2 loss or knockout, are you guys seeing any indications that CRISPR-Cas9 pathway is being elevated?
So we haven't looked at it, but what happens when you have telomere fusions is that you induce such a genome instability that you're going to have chromosomal DNA and telomeric DNA that is going to exit the nucleus. And when we do this telomeric fusion assay, we really need to induce TRF2 depletion and look within 5 days at a moment when we do see the fusions, but the cells are not completely dead yet. If we let them go, they are going to die, and that is going to likely be dependent on the CRISPR-Cas9 pathway.
Great. Thank you for your excellent talk.
So I have another question for you, Nausicaa. So, PARP inhibitors, at least from my rudimentary understanding, that they in part work almost, it's not just that they're not really inhibitors. They're sort of creating a sort of a frozen state. In fact, I think resistance to PARP inhibition, again, Ben will know way more than me, but can be mediated by losing PARP.
So that is true in HR proficient cells. That is not true in HR deficient cells, and usually people don't make the distinction. But it's very clear in our screen, for example, we did all these cell lines with PARP inhibitors, and what rescues PARPs in HT1080 when you add PARP inhibitors is suppression of PARP, of PARG, and all this pathway that rescues. In BRCA1 or PALB2 cells, this is not true.
Okay.
Yeah.
That's good to know. All right, thanks. So any other questions for Nausicaa? We had a lot of great discussion there. And if not, let's all thank Nausicaa for a fantastic talk, and hopefully there'll be some great collaborations that come out of this. All right, thanks everyone. Nausicaa, thanks so much, and have a great rest of your day.
Thank you. Thank you for being here.